Okay, so once again, just quick reminder, if you decide to use your voice, it will be recorded, so heads up. So let's take these questions one by one, and what I would like to have happen is for you guys to try to answer them, and then we can kind of talk through them. So I'll read you the first one, and then you can tell me what you think. So which of the following would have the most profound, parentheses, serious effect on sperm production? And then we've got low LH, low T, low GnRH, and then finally higher than normal testicular temperature. So what do we think about this one? So someone in the chat, also quick programming note about recording. For when I talk about people's responses in the chat, since we are recording, to keep confidentiality, I'm not going to say anybody's name. So if I don't say your name while I'm addressing your answer, I'm not being rude. I'm just protecting your right to not have your name on the internet. So one person is saying GnRH. One person, two people are saying higher than normal testicular temperature. So I made this question this way on purpose, and here's why. A lot of nursing or qualifying exams for other types of clinical professions, so like the NCLEX and such, you're going to be faced with questions like this. They are nobody's favorite question, but they do happen. And so I feel like I need to help prepare you for them. The questions are ones for which a lot of the answers, even all of the answers in the choice, are somewhat correct, but you have to pick the most correct one. It's a really annoying type of question, but it's also really powerful for assessment of mastery. So students don't like them, and I totally understand why, but they're a necessary evil. So your job and my job is to work on preparing you for the eventuality of these questions so that you feel okay facing them later on in your careers. So, is D incorrect? It does have an effect on sperm production, but notice that I'm using a superlative here. So most profound or most serious. So yeah, it does have an effect on sperm production, but ignore all these other choices for now and think about if this was only happening and everything else was normal. You still had normal hormone levels, you would still have okay sperm production. The thing that would be affected would be the quality of the sperm produced. So tempting answer, not correct. And then we have some hormone choices. So here I've used the abbreviations for the hormone. And as I mentioned in a previous Zoom, I would really, really encourage you to learn what these abbreviations mean. It's a step that a lot of students don't take and end up regretting later. So while it says LH up there, I'm going to say luteinizing hormone. So A is low luteinizing hormone, B is low testosterone, and C is low GnRH. So I will tell you right now that the correct answer is low GnRH. Does anybody care to take a stab at why that is? I will say that in a moment. I just want to give you guys a chance first. Bingo. Great answer. So the answer that came into the chat said, it is the start of the process for all of the other hormones. And then another person is saying that's what stimulates FSH and LH that targets the testis. So, and then another person is saying it initiates the process. All of those are correct. They're just different ways to address the same idea, which is that GnRH, gonadotropin releasing hormone, is the hypothalamic releasing hormone that causes both follicle stimulating hormone and luteinizing hormone to be released from the anterior pituitary. Those are what initiate sperm production, and those are what carry it on. So let's draw a little arrow to here and say low GnRH, so causes release of FSH. And FSH, follicle stimulating hormone, essentially stimulates the nurse cells in the testes to nudge the spermatogonia to do mitosis. So let's put and then LH acts on interstitial cells to cause T production. So luteinizing hormone is related to testosterone because it causes testosterone production, which happens in the interstitial cells. Those are the cells that produce T that are between the seminiferous tubules. And so that keeps androgens locally very high in the testes. So great job on this first one. Okay, what do we think the answer is to number 2? Coagulations, that should be just coagulation. Okay, so in the chat, I am seeing 2 people saying vascular spasm, C, and that is the correct answer. So this one's a little bit more straightforward because there's some ones that are obviously wrong if you know a little bit about coagulation, right? For example, this is the last step. This is sort of the big bang at the end of the 2 clotting pathways because you don't get a clot without fibrin. So that's obviously out. The only other kind of tempting one is platelet plug forms, but that happens after vascular spasm. So this is narrowing of a broken blood vessel. The idea here is really, really simple. All of your blood vessels are tubes, right? So you've got tubes with blood in them. If you have fluid coming out of a tube, one good short-term way of making that happen less is just to make the end of the tube a lot narrower. And that's what vascular spasm does. So that is the correct answer. Very, very good. Interesting side fact about this, not something I would test you on, but something that's good and interesting to know. You guys have learned about serotonin, or you know about it in your regular lives. Serotonin is a hormone that's, or not hormone, neurotransmitter, excuse me, that's commonly associated with mood. So like if you have clinical depression, you'll take something that elevates your brain serotonin. Serotonin does about a bajillion things besides regulate your mood, and one of them is it contributes to the vascular spasm step of clotting. So if you have really, really low serotonin globally, like not just in your brain, but everywhere, that can manifest as problems with blood clotting. It's difficult to do that. It's not a common pathology, but it does happen. There's a very dangerous condition called serotonin syndrome, which results in globally depleted serotonin, which can have that effect. Okay, so number 3. This one I also made a particular way on purpose to demonstrate a particular problem that comes up for some students on exams. And that is, and I'm seeing at least one correct answer here. So let's talk about those in a moment. This is what I mean when I say I might not ask you about the definition of something, but you need to know it in case I use it in the wording of a question. So let's say you were aware of the uterus, but you compartmentalized this word as being for lab only, and you didn't regard it as a term for lecture. You still need to know the definition of this word because you need to be able to use it to understand this question. Note, I haven't asked you to define myometrium anywhere, and I won't, but rather you need to make sure that you know which layer of the uterus I mean, and that this word is associated with the uterus to get this question right. So that's the reason that vocabulary is important, not because I'm going to sit here and say, define myometrium, define parametrium, but rather because I'm going to use those words, and if you're not solid on what they mean, you won't be able to answer the question correctly. So we've got some answers. Let me have a look. So although estrone is a hormone that is one of the quote unquote female sex steroid hormones, it's not actually one that we really talk about in the book all that much. So we're going to leave that one off. Also, what is the myometrium made of? That's another key here. Who can tell me? Smooth muscle. Perfect. Very good. Yeah. So the myometrium, the prefix myo means muscle. It doesn't specify what type, but if you've done your reading, you'll know it's smooth muscle. So the only one of these choices that has a profound effect on smooth muscle is oxytocin. So D is the only choice. Now, if I replaced myometrium with endometrium, then you'd really have your work cut out for you, right? Because you'd have to ask yourself whether estrogen or progesterone had a stronger effect. Quick hint, the answer is progesterone. But I said myometrium, so this is the only candidate answer. So let's fill that in. I'm going to say all smooth muscle. I'll call it there. What I was going to type after that was that expresses receptors for it. Okay, awesome. We're really moving along here. I'm pleased about that. So for if the hypothalamus wants to signal the anterior pituitary, it uses blank signals, whereas if it wants to signal the posterior pituitary, it uses blank signals. So this is one of those pesky ones where you have to fill in 2 blanks and put them in order. So take a moment to tell me what you think, and then we will discuss. Okay. The correct answer is B. So I made this question for this review specifically because this topic has been coming up over the past several Zooms, which tells me that A, you guys are curious about it, and B, people who are attending Zoom don't feel 100% confident about it. And I would like for you to feel confident going into the exam. So I thought this would give us an opportunity to sort of practice with this idea. So this is about that bullet point on the study guide that's talking about communication between the anterior and posterior pituitary. The modality, meaning the kind of communication that is used, is not the same between anterior and posterior. So the correct answer is humoral, neural. So humoral means in the blood. So I'm going to put in parentheses after this. I'm going to abbreviate hypothalamus. So releasing or inhibiting hormones from hypothalamus travel to the anterior pituitary in the blood. Now the second one is neural because the cell bodies that produce oxytocin and antidiuretic hormone, those cell bodies are actually up in the hypothalamus. So they're producing those neuropeptides, and then the neuropeptides are carried down the axons of those neurons. The axons of those neurons pass through the infundibulum of the posterior pituitary, and the axons terminate in the posterior pituitary body, the pars nervosa. So all the hypothalamus has to do when it's time to release oxytocin or antidiuretic hormone is fire off an action potential down those neurons, because oxytocin and ADH are sitting in the end of those axons, and they are released in the same manner that any other neurotransmitter is released via exocytosis in response to an action potential. So that's what I mean when I say neural, an action potential is involved. Does that make sense to everybody before we move on? Okay, good. And 5, finally. So atrial natriuretic peptide, or ANP, is an antagonist of which hormone? What do we think? Awesome. I'm seeing some correct answers right off the bat. That makes me really happy. That helps me feel confident that you guys are ready for the exam, which, believe it or not, is what I want. I don't take an adversarial position against my students. I actually want you guys to be successful. So I'm stoked that we are talking about this. So the other possibility is ADH. The answer is actually aldosterone. This is an example of a question that's a little bit harder than one that I would probably put on your exam. But I put it on here because I wanted to sort of help you guys prepare for this idea. So it's kind of a reach question, put it that way. So this requires that you would have read a little bit into the mechanism of action of these hormones, which does exist in your book. But this is a pretty heavy duty question. So let's first eliminate the ones that are obviously wrong. So not related to water balance in this way, really at all. And glucagon, this is involved in glucose homeostasis, not water. So those ones are both out. So the answers I'm seeing in the chat are either D or A, which means that you guys have arrived at the sort of problem area of this question, which is if you know your hormones well enough, you'll be like, okay, so ADH, antidiuretic, so it's against diuresis. And I know that ANP causes diuresis. So A is correct-ish, but it doesn't directly oppose the action of it. So here's the deal. So it does prevent diuresis, but the crux here is the way that it does that. So I'm gonna actually give myself some carriage return so we can have some space to work this out down here. So atrial equals comes from atria of the heart. So this is one of those hormones that is being released by an organ whose primary function is not endocrine. So remember on the study guide, I asked you guys to be able to name at least one hormone that comes from, you know, organs that are not classically endocrine. So by that, I mean the heart, the kidneys, adipose tissue, the stomach, etc. So ANP is the one for the heart. And then natriuretic. This one is tricky. Who can tell me what that ion is? What does Na plus stand for? Sodium, very good. Yeah, so here's the tricky part of ANP. The Na in natriuretic refers to sodium, but we're using the chemical name as part of the word. So anything uresis is loss of something in the urine. So diuresis is loss of water in the urine. Natriuresis is loss of sodium in the urine. And in this case, at the expense of potassium. So ANP acts on the sodium-potassium pump to change the way it works and change the exchange between the two. So you might be wondering, well, why does loss of sodium in the urine mean loss of water? And the answer is osmosis. So the rule of osmosis water follows solutes, but in this case, it especially wants to follow sodium. Sodium as an ion is a very attractive ion to water. So basically, if you're peeing out a bunch of sodium, water is going to follow it. So atrial natriuretic peptide causes a loss of water and therefore blood volume by making you pee out more sodium, and then the water follows suit. Aldosterone does the opposite. So aldosterone and ANP are antagonists because they do the exact opposite of each other. ADH also prevents diuresis, but it does so via a different mechanism. So if you're thinking back to the renin-angiotensin-aldosterone system feedback loop chart, you'll notice that there's a little box and it says in it that aldosterone and ADH are released in tandem with each other. So if we know that aldosterone does that and that antidiuretic hormone does this, those two things together are a very, very, very powerful way to rapidly make sure that you are not peeing out water, which conserves water very strongly. Another consequence of ADH release, not so much aldosterone, is that you get the sensation of being very thirsty, which causes you to selectively consume more water. So as you might imagine, if you're really conserving water by not losing it in your urine and you're drinking a bunch more of it, that's a very good way to get your blood volume back up pretty quickly, right? So, we have any questions about this last one? It's okay if the answer is no. I just want to check before moving on. Okay, awesome. Thank you. Hopefully that kind of clears up those relationships. I know it gets a little bit messy, especially when two things do the same outcome, but in different ways. So I am going to not get rid of these. I'm going to save this. You'll have it on recording, obviously, but I'll also save it and post it just for help purposes. So it'll be up under the supplemental material heading. Yes, here are the first two questions. But again, I'll post these, and there's going to be a recording of this. So you'll be able to see it again at any time. You don't have to scramble to write it down if that's what you're doing right now. Okay, so hopefully that served as a good warm-up for you guys. You're welcome. So if anybody has any specific questions, I'm happy to address them. So go ahead and type them in the chat if you do. If not, today, even though we're moving the exam a little bit, today is technically the due date for the case study. And so I thought a good use of this recording time might be to go over the case study answers and have a little discussion about those. How do we feel about that? Is that something that you guys want to do? Okay, I figured the answer would be yes, but it's your time, so I wanted to make sure that you wanted that before I just hauled off and did it. So let me go find that, and then we can talk about it. I will say, while I go through and locate this thing, that the ones that I have graded so far have been really good overall. I was very impressed with how well students were able to do that assignment. So good job. Yes, it is, but we're kind of up against the wire for the test, and I want to make sure that everybody has those sort of concepts down before the exam. So I'm okay with going over the answers now. Looks like I'm gonna have to hunt it down in Canvas. It's fallen out of my, oh wait, no. If you're watching the recording of this in the future and not right now, I apologize for the dilly-dallying. It is due tomorrow. Okay. Yeah, it is due tomorrow, but since we're up against the test and the whole point of the case study is to prepare you for the test, I figure we might as well look at it right now. Okay, so let's start with the rat assignment. And so there's a question in the chat, and I'm going to read it for the recording because it's a good one, and I'll explain why after I read it. So the question is the following: what does the thymus have to do in the rats? I thought the thymus is bigger when you are young and shrinks with age. I'm having problems with the different sizes in rats and the cause of it. Yes, absolutely. The thymus size change is one of the sort of confounding findings in the rats. And so let's discuss the thymus. The person who asked that question made the correct observation that it's bigger in childhood and infancy and smaller with age. But that's just a phenomenon that it exhibits. That's not what it does. Can anybody tell me kind of what the thymus does? So it releases two hormones, thymosin and thymopoietin. So if you weren't sure about the thymus on this case study, what would be the best to do would be to go look up some info in the thymus and find out what exactly it does besides just getting smaller. It does that, but that's not its primary function. So the primary function of the thymus is to develop and help maintain the immune system. So a change in the thymus in this exercise doesn't necessarily mean that the thymus is being directly manipulated with hormones, but rather Any hormones that cause the rat to feel stress are likely to cause the thymus to shrink, because if you read about the stress hormones, things like cortisol and corticosterone tend to cause loss of body mass and depressed immune system. So if you see a skinny rat with a smaller than normal thymus, that's telling you that there's something up with the immune system. Does that make sense? Okay, so with that, let's look at rat A, because that kind of relates to what I'm talking about. So I'm actually going to go in and type in the text boxes, because up here I provided you with a little chart where you're supposed to, or I guess it's down further, where you're supposed to put pluses and minuses, but I'm just going to do it in the boxes because it makes more sense to do that here since we've got the control rat right here. Okay, so body weight, negative, and I'm going to put 2 minuses because it's only 100 grams, which from a human perspective isn't that much, but if you're a 300 gram on average rat and you lose 100 grams, that's a third of your body weight, which is pretty big. So double minus, the testes lost weight. The prostate gained a little bit of weight, but only 5 milligrams. So I'm going to put trace, meaning it changed a bit, but small enough change that it's not remarkable. The adrenals more than doubled in size, so that's a big plus. Seminal vesicle lost weight. The thymus lost weight. I'm going to put 2 minuses here because 475 down to 250 is quite a big drop. The thyroid lost a little bit of weight, but I'm going to say not that much. And the pituitary lost weight as well. So if I was going to summarize this rat, I'm seeing skinny rat with depressed immune system and failure to maintain sex organs. Now, all of these results are kind of screaming stress hormones, right? You guys can kind of see that, hopefully. Here's the deal, though. You have to figure out which level of the feedback loop is being messed with. So you have some choices now. Once you've noticed, like, okay, this seems like a stress hormone related thing, then you have to establish, okay, well, is it a hypothalamic hormone? Is it a pituitary hormone? Is it an adrenal hormone? Which hormone am I introducing into this rat? Keeping in mind that when you introduce a hormone from the outside, whatever gland normally produces that hormone tends to shrink. So let's start with that idea. It would be tempting to look at this and say, the rat was injected with cortisol. Why is that not true? What specific organ change tells us that that's not what it is? And I'm waiting just to give people a chance to type. I don't know is also an acceptable answer. Thank you for saying that. I appreciate that because it helps me not pause for too long. So saying I don't know is also great, you guys. Don't hesitate to do that. I appreciate it. Okay, it's okay not to know. So here's the deal. If it was cortisol or corticosterone, those are both hormones that come from the adrenal cortex, right? So if I was injecting cortisol or corticosterone into this rat, I would expect to see the adrenals lose weight, not gain it, and certainly not double in size. So it's for sure not the adrenals, because the adrenals did not lose weight, they gained weight. So we saw a pituitary gland that decreased in size. What stress-related hormone does the pituitary gland produce? ACTH is correct. Very good. So that's adrenocorticotropic hormone. So the correct answer for rat A, and I'm not going to give you the answer to every single rat here, but I am going to help you work through some of the trickier ones, because I really want you to see how this is done and feel confident in doing it. So adrenocorticotropic hormone, corticotropic means causes the release of cortical, meaning cortex-based hormones. So adrenocortico means adrenal cortex. So the pituitary releases ACTH to cause the adrenal cortex to release stress hormones. So the reason the pituitary has lost weight is because if I'm injecting ACTH, I've effectively relieved the pituitary of having to do that. So it's going to lose a little bit of mass, because the cells in there that are making ACTH, their job is done. They don't have to be maintained as much. So we see this slight mass drop in the pituitary, but I'm also giving the rat a supraphysiological dose. Supraphysiological means bigger than the rat would produce on its own, dose of ACTH. So I'm really stimulating the adrenal glands hard. Overstimulated glands tend to hypertrophy or increase in size. So the only explanation, even if I didn't really look at the rest of these, if I saw a huge positive mass change in the adrenals and a drop in pituitary mass, that tells me that it's ACTH. So this information alone, adrenal and pituitary, would be enough to tell me that rat A was injected with adrenocorticotropic hormone. So this is why this exercise is powerful, because it forces you to examine every piece of the feedback loop. So does anybody have any questions on rat A or how I did that before we move on? And the answer no is also fine. So if you don't have any questions, we can just continue. Okay, so I'm seeing no's, good. Second question, does what I just showed you make logical sense? Okay, cool. I'm seeing yes. Somebody in the chat is also saying they did get ACTH, but still stuck on thymus. So the thymus is responsible for immune system maintenance. If I'm causing the adrenals to increase in size by stimulating them with ACTH, what does that mean that they're releasing? Probably stress hormones, right? So the piece of the puzzle that I'm not showing you here, I'm just showing you change in organ mass. But if the adrenals are hypertrophying and I'm bombarding them with ACTH, odds are they're releasing cortisol and corticosterone. Those are stress hormones. Chronic stress hormone release causes immune system depression. So the direct consequence of immune system depression is the organ that helps maintain the immune system is going to get smaller. So it's not that I'm injecting any hormone that takes the place of the thymus, but rather it's just a consequence of having a sustained stressor on the rat. Does that make sense? Good, okay. Alrighty, so that's kind of how you do that exercise. The whole thing about getting kind of hung up on the thymus is something that happens almost every time I give students this cognitive task. So If that's you out there, and I'm sure that there's more than one of you, don't feel bad. It's totally normal. This is an assignment that I typically give my upper division behavioral endocrinology students, and I make them do it for a grade. I don't make you guys do it for a grade because it's kind of a reach assignment. So it's one that's normally assigned to 400 level students. So the fact that you guys are doing so well at it is really impressive. Good job. Okay, so I'm only going to do rats A and C for this one because I don't want to give away every single answer since it's not quite due yet, but I wanted to help you guys flesh out sort of what's going on and how to sort of tease out these answers. Okay, so this one's going to be tricky because I have to scroll back and forth, but we've got, I'm going to say plus plus for body weight. So we've gone from 300 to almost 500 grams. So look, the testes, 3,200. Oh, we got a testes mass drop. So let's put minus here. I'm going to say minus minus. Prostate is big plus. Adrenals is trace minus. Not trans, trace. There we go. Seminal vesicle, we can see, has more than doubled in size. The thymus has experienced trace minus. Thyroid is trace plus. And let's check out the pituitary. So this one is also a little bit tricky because there's two tempting answers. So if we go through and just kind of look, we've got a really big rat. And the big rat has lost testicular mass. But it's got really big other secondary sex characteristics. And then we've got some trace changes in other ones here. So what do we think for rat C? I'll give you guys a minute to think about it. All right. So I'm seeing LH and testosterone. So those are the two candidates. Yes, indeed. So now we have two competing theories, and that's one of the reasons why this one is tricky. So I'm trying to find something to illustrate my point here. So just give me one moment. And while I find this thing, why don't you guys look at the data here and think about those two answers and which one is more likely given that scenario. Okay, so I'm not finding the thing I wanted to, so in the interest of time, I'm just gonna go ahead and go forward with this. So we have a slight pituitary mass change, which is a little bit tricky. So now we have relative magnitudes to think about. So the major clue anyway, is testes. And so if you're trying to juggle around in your mind and figure out, okay, is it testosterone or is it LH? That is a tricky one. And it's tricky on purpose because the feedback loop here is complicated. If you go to the male sex characteristic feedback loop in your book, which I'm actually getting right now. What you'll notice is that the regulatory pathways that allow the gonads to talk to the pituitary and to the hypothalamus aren't necessarily the most straightforward. So when you're trying to make this decision, your job is to look at testes and pituitary and say, okay, so which of the changes in this case has the greater magnitude, and what do I think that means? So I'm trying to turn to this page really quick. There we go. Okay. So before I answer it for you, oh, Heather, I am turning to page 1071 in the book if you want to follow along, because the answer is on this page. I just wanted to be able to describe it to you. So if you're following along, notice we have a slight pituitary mass loss, but a much larger testicular mass loss. So have a look at page 1071, specifically looking at negative feedback. And what you'll notice is that testosterone feeds back negatively on GnRH. So imagine I take a syringe and every day I go to this little rat and I inject it with a bunch of testosterone. What that's going to mean is that in the long term, gonadotropin releasing hormone is going to be inhibited, which means that I'm going to be preventing the release and secretion of FSH and LH from the pituitary. So the trace pituitary mass loss that we see isn't because I'm replacing the pituitary's function, but rather because I'm chronically inhibiting it, meaning I'm preventing it from doing its job, part of its job anyway. Of course, it's probably still releasing the other regular tropic hormones, but I'm not letting it do the FSH and LH stuff. So that accounts for the slight pituitary mass loss. I've taken the place entirely of the testes by releasing testosterone, which is why we see that very profound testicular mass loss. So LH is a tempting answer. It's not quite the right one for the reasons I just described. So the best answer for rat C is testosterone. If you are following along and looking at page 1071, does that make sense based on looking at that feedback loop chart? Awesome. Okay. So yeah, this exercise is really all about feedback loops and using information in these little boxes as well as your understanding of the feedback loops to sort of make decisions about which hormone is being administered. So that was sort of a detailed dive into how we tease apart this information. So let's move down to the blood section. I just did it by scrolling back and forth through the boxes, but if you want to do it a little bit more easily, you can use this chart here if you haven't done so already. So who can tell me what the answer to A is? Very good. Yes. So someone in the chat is answering A, B. And you can tell that because there are no anti-A or anti-B antibodies in the patient's plasma. I'm very pleased to note that no Rh type was included in that answer. So one error I often see in students' answers to part A that tells me they don't understand Rh fully is their answer includes something like they are AB negative because something something antibodies. So these data only tell you about the antibodies in their plasma relating to their ABO blood type. It doesn't have any information about the Rh factor at all. And that is the problem. So I ask, what went wrong with the first transfusion? Explain your answer. So we've had enough time for you guys to look at this assignment and think about it. And I didn't give you the answer in previous Zooms because I didn't want to give it away, but we're almost there now. And I think it's fair to work through it together, especially since y'all bothered to show up for Zoom today. Good job. So I would ask you, but it'll take too long to type. So the sort of Cliff's Notes version of what went wrong with the first transfusion is if you read through this and you notice that the Rh factor was never mentioned, that's your red flag right there, right? So if your nurse is doing their job correctly, they're going to report the ABO antibody serology as well as whether or not any Rh factor antibodies were found. And since they didn't, the conclusion that makes the most sense and is the most parsimonious is exactly what you just said in the chat, which is that the patient was AB negative, but because the positive negative was omitted from the serology report, they got AB positive instead, right? So that's the answer to B. So I end up getting a lot of emails from students about this exercise being like, I can't answer the question because it doesn't have the Rh factor in the little blurb at the top. And that is the point, you guys. So that's the answer to that. And then if we know that they're AB negative, then you can easily answer to whom they can donate and from whom they can receive. Does what I just said make sense? Awesome. So what went wrong with the first transfusion is that they got the wrong Rh type of blood. So the Rh factor was never mentioned up here. And so you can assume that the doctor, you, in this blurb, transfused with AB blood, but you gave them Rh positive blood when they were Rh negative. So let me ask you this. Would the answer to B be the same if the patient was Rh positive and you gave them Rh negative AB blood? It wouldn't, exactly. So that's the part of the Rh factor that's confusing for a lot of students, and I understand why. It's not at all intuitive, is that the Rh factor antigen antibody relationship is one-sided, meaning if you're Rh negative, you cannot get Rh positive blood because your antibodies in your plasma are going to attack the donor red blood cells. But if you're Rh positive, you can get Rh negative blood because you don't have any antibodies against the Rh factor in your plasma to begin with, and the blood cells you're receiving don't have any Rh antigens on their surface, so you would be okay. So there's a little bit of an asymmetry in the transfusion reaction equation there. Okay, so that is case study number one, tips and tricks and such, not all of the answers, but hopefully that helps sort of clarify some insight into how you can successfully complete that assignment, bearing in mind that I'm not grading it for whether or not you get it perfectly right. I've already graded a couple who got some of the rats wrong, and they still got 10s because they still showed effort. So you do not have to be correct. You just have to show that you made a good faith effort to try and to explain your answers. Okay, so we've done the two things I wanted to do this morning. So now I'm gonna open up the Zoom for questions, and I'm gonna continue recording for this. So if you have questions and you don't want to use your voice, just go ahead and type them in the chat. I'm gonna give you guys a few minutes to do that. Okay, we've got an excellent question in the chat, and that is about clot retraction. Yes, it is part of it. And then we'll go over this other question next. So let me pull up a couple PowerPoints to point at while I talk about these two questions, and we will get going. Alrighty. So I'm just going to run through these really quick, try to find the slide that I want. So unfortunately, I don't have a good picture of this because your book doesn't have one, and I wasn't able to find one on the internet that wasn't watermarked. But it is indeed part of the coagulation phase. So the idea here is really bullet point number 3, which is that you want to reduce the size of the damaged area. So a good analogy for this is, if you're bleeding, the implication is that there's been some tissue damage, right? So usually if you're bursting a blood vessel, that means something has messed up your tissues. So you're going to have some ragged edges. One of the cool things about clotting is that not only can it patch a hole, but imagine you have like a rip in your jeans, right? And you want to close that rip. You're going to pass some thread through the edges, right? And then you're going to pull on the thread, applying tension, and that's going to draw the edges of the rip together. And then you can go ahead and tie off the thread, and voila, your jeans are fixed. Clot retraction operates on the same principle. So basically, you're going to want to draw cut edges together and reduce the size of the damaged area. And that is part of the coagulation phase. Does that make sense, person who asked that question? Awesome. Okay. Did I answer everything you wanted to have answered before I move on? Cool. Okay. Then let's move on to primordial follicles. Okay, so the question is for the recording and for everybody, can you go over the decreasing amount of primordial follicles as the months passed? Yes. In order to address that, I have to go back to a time before monthly cycles. And this is an important difference between males and females. So notice, if you're careful, you'll see that, and let me grab my laser pointer here. You'll see that birth and fetal development are mentioned a couple times here, and that's because a lot of the preparation for oogenesis happens before birth. So, oogonia are stem cells. They're analogous to spermatogonia. By the time someone is born, they don't have any oogonia left. So that means there are no more stem cells. You can't make any more primary oocytes. By the time you're born, you have all of the ones you're ever going to have. Right? So between the 3rd and 7th months, so once you're done making all your primary oocytes, then those primary oocytes are permitted to enter meiosis 1, but they just stop. So between birth and puberty, in a child who has ovaries, they have this big, huge cache of primary oocytes. A primordial follicle contains a primary oocyte. So the immature ovaries are just sitting there with this big stash of primary oocytes housed in primordial follicles, waiting. Once puberty happens, we get this increase in FSH, and that triggers the start of the ovarian cycle. So each month, once that happens, we get the increase in GnRH pulses, and that stimulates secretion of FSH, follicle stimulating hormone. And in that case, not just one, but a few of the primary oocytes in an ovary are stimulated to develop further. So you've got this handful of primary oocytes developing each month. Usually only one of them makes it to ovulation. The ones that didn't make it to ovulation undergo a process called atresia, where they just stop developing and die. But because you only have a fixed number of primary oocytes, let's go to the next slide, you have a fixed number of these, every time you use some up, so ovulate one, the other ones go atretic, you've depleted your supply a little bit because you can't form new ones. So whereas sperm production goes on for as long as hormones are in place, so as long as the male is producing gonadotropin releasing hormone, sperm production will continue because there are stem cells. With oogenesis, the same is not true, because by the time you're born, your stem cells to make more gametes are all used up. You don't have any. And so you're just drawing on an existing supply that you're depleting a little bit every month. Does that answer your question? Awesome. I do want to touch on one cool thing before we move on from this topic, if we're going to. So, and not to do this, I'll use Epic Pen and draw it. So I'm going to draw this, but I have to switch my screen share to do it. So just bear with me. There we go. Okay. So here's something nifty about that particular thing that a lot of students don't think about, but it's kind of amazing. And it is this. And I'm going to draw it out, so I'm moving my pen around here. So I have, well, let's see, how am I going to draw this? So let's say we have a pregnant lady. So this is going to be me trying to draw a pregnant lady. Prepare for bad drawing. So we got some pregnant mammary glands. And wait, go back. Big old belly. There, pregnant person with some legs. And I'm not going to draw their upper body because I don't need to for this. So this person has ovaries, which I'm going to draw them being pregnant. So I'm going to push the ovaries to the side. So out of one of those ovaries, spraying our fetus, which I'm going to draw in here. And let's say the fetus happens to be female. So the fetus also has ovaries. And I'm going to draw the fetus in the breech position just because it's really hard to draw fetuses upside down. So we've got some little arms, a little head, a little shut eyes, a nose. Here's our little fetus. Little feet. Okay, there's my little fetus. So if the fetus is female, remember between the 3rd and 7th month of development, their primary oocytes are sort of finishing up, meaning that they're making all of the gametes that they're ever going to have in their life right now. So if I zoom in on this, we've got primary oocytes forming. So here's the cool thing. So I'm genetically and physiologically female. I have ovaries, and you know, as is tradition, so did my mom and so did my grandma. So one of these primary oocytes was destined to be me. And it was inside of my mom when my mom was forming. Inside of my grandma. So although I did my major cooking, so to speak, inside of my mother, when she was pregnant with me, I formed when my mom was a fetus. At least half of my genetic info did. While my mom was a fetus, still inside of my grandma, which is pretty crazy if you think about it. So that is my wow moment for the morning. Technically, in some sense, when your grandmother was pregnant with your mother, you were in there, or what would eventually become you anyway. Neat, huh? Also, that's where all your mitochondria come from, and that's why mitochondria are matrilineal, meaning you only get your mitochondria from your mom and your grandma and so forth, not your dad. Do I have any questions about that or any more questions about eggs? So I'm going to go back to sharing my other screen here. So regarding the part of the question I was answering where I talked about atretic follicles, atresia is basically the developing follicles that don't end up being ovulated. They undergo that process called atresia, where they basically just stop developing and end up undergoing apoptosis. So they just degrade and they're gotten rid of. It turns out that if you look at the follicle development over a cycle, what you end up with is different percentages of follicles going atretic depending on which stage you're in. So, for example, it seems to decrease initially and then once again increase. So of the follicles that make it to class 7, which is right before ovulation, about half of those end up not actually being ovulated, and 77% of class 6 ones end up not being ovulated. So that's what this chart means. Okay, we've got another question, and that's about hemolytic disease of the newborn. So let's go back to that PowerPoint real quick. I said escape. There we go. Okay. So before we even talk about RH types, let's just talk about placentas. Oops. So placenta is an organ that is grown during pregnancy, and it is a handshake agreement between the fetus and the mom. So both parties participate in constructing the placenta. And the placenta does a lot of things, including make hormones and do other stuff, but it also functions to help the maternal blood exchange substances with the fetal blood to keep the fetus alive. So we want oxygen-rich blood going to the fetus from the placenta and oxygen-poor blood going from the fetus to the placenta. But it's important that during the pregnancy, the two bloods don't actually touch. So we have blood vessels that are in close contact with each other. So they're very, very nearby, but the physical blood is not mixing. And this is only a problem when mom is Rh negative and fetus is Rh positive. So if we have a mom carrying their first Rh positive fetus, that means that we don't know if the Rh negative mother has been sensitized to Rh positive blood or not. In most cases, medically, if your fetus is tested and known to be Rh positive and you are Rh negative, you are given a drug to prevent hemolytic disease of the newborn, regardless of if there's serum antibodies in you, the pregnant person, or not. It's kind of a better safe than sorry principle. Sorry, I'm getting my laser pointer. So I'll talk about ROGAM in a little bit. But essentially, if there is Rh negative blood cells in here, we just kind of assume that there's antibodies before, but this is the actual process. So while the blood supplies are separate during gestation, delivery is messy. If you've ever had a baby or watched a baby be born, you know that, right? There's a lot of goop, there's a lot of blood, lots of blood. And part of the reason for that is that the placenta tears off the endometrium and is passed out of the vaginal canal after the fetus, typically. So when the placenta tears off, blood vessels get broken and fetal blood ends up mixing with maternal blood. So if the fetus is Rh positive and mom is Rh negative, and if mom has never been exposed to Rh negative blood before, or Rh positive blood before, excuse me, after birth, the immune system is going to get to work making antibodies against the Rh positive blood that it encountered during delivery. So it's the same principle as any other antigen sensitization, where you get exposed to something and then your body is like, I want to remember that for next time. So I'm going to make some antibodies against that thing for next time. So if you read the box here, it says about 20% of Rh negative mothers who carried Rh positive fetuses became sensitized, meaning developed anti-Rh antibodies within 6 months of delivery. It takes time to build up an antibody defense this large. So what that means is, although the antibodies end up being made, there are too few of them and they happen too slowly to affect the first fetus. Now, let's say mom becomes pregnant again, mazel tov, and again, the fetus is Rh positive. During the second pregnancy, mom has had time to make anti-Rh antibodies. Red blood cells are too big to cross the placental barrier. So Rh negative cells can't get in here, Rh positive cells can't get in here. Antibodies, though, antibodies are small. They're just proteins, and they can cross this barrier. So hemolytic disease of the newborn happens when anti-Rh antibodies from mom enter the fetal blood supply and begin attacking the fetus's blood cells, causing them to agglutinate and eventually lyse. And this is obviously very dangerous for a newborn because it literally means that their blood is exploding and turning to goop. Not ideal for a neonate. So that's what hemolytic disease of the newborn is. However, we have a drug for that now. It's called RhoGAM. And basically they're antibodies that are specifically in place to destroy fetal red blood cells before any that cross the placenta before they can stimulate an immune response. So RhoGAM, the drug, is actually built using special antibodies that only some people have. And so we extract the antibodies from those people and use their blood to synthesize this drug. So it's something that was discovered already existing in humans rather than something that was invented. Does that answer your question? Awesome, good. All right, any other questions? I'm happy to answer them if we do. If not, or if nobody has anything pressing, I'm okay with signing off early also because I need to go exactly what you're saying in the chat. I need to figure out the reproductive system part 2 problem, which takes some doing. So that's next on my list of things. In the meantime, though, I'm going to stop recording.